DNA is the largest macromolecule represented as genetic material of the cell. It is one of the two nucleotides present in all living organisms except certain virus. It is present in linear form in nucleus and in circular form in mitochondria and chloroplast and make them semi-autonomous organelle in the cell. It is commonly known as informational molecule because it stores and transfer the information needed for the specific protein which will determine the structure and function of the cell. Hello everyone, I am Suchi. Welcome to my channel. Let's discuss today about DNA. DNA or deoxyribonuclic acid. This DNA normally contains one pentose sugar, nitrogenous base, and phosphate group. There are two types of pentose sugar present in cell. One is ribose, other one is deoxyribose. This ribose is C5H10O5 and this deoxyribose is 6,5H10O4. By seeing this name, you can also understand one oxygen is absent in case of deoxyribose. If I will make its structure, it will be like this. This is the structure of ribose, but at the position of C2, OH is present in case of ribose, but in case of deoxyribose, at this position you will find H group instead of OH. So in DNA, deoxyribose are present as pentose sugar. So this will be the structure of deoxyribose. Next, these nitrogenous bases are heterocyclic compound. It contains ring-like structure. It mainly contains carbon and nitrogen. There are two types of nitrogenous bases are present in cell. One is purine, other one is pyrimidine. There are two types of purine bases are present. One is adenine, other one is guanine. And there are three types of pyrimidine bases are present. Those are cytosine, thymine and uracil. But in DNA, adenine and guanine present as purine base, cytosine and thymine present as pyrimidine base. In DNA, uracil is absent. This uracil you will find in RNA. If I will make its structure, this is the structure of adenine. This is the structure of guanine. These are the bases of purine. This is the structure of thiamine. And this is the structure of cytosine. So this adenine, guanine, cytosine and thiamine are present as nitrogenous base in DNA. Then this phosphate group, which is PO4 3- is formed from H3PO4, or you can say phosphoric acid. By dissolution of this triple hydrogen, it will form this phosphate group. This phosphate group will bind with this nitrogenous base and pentose sugar, and it form the nucleotide. Due to the presence of this phosphate group, it make this nucleotide is acidic in nature, which will bind with other basic protein and cation. When this pentose sugar make a bond with this nitrogenous bases, it will lose water and it will form glycosidic bond. And this pentose sugar and nitrogenous base combinedly form nucleoside. And this side, when it will make a bond with this phosphate group, it will form another one covalent bond, that one is ester bond. And combinedly they will form nucleotide. DNA is a double helical polynucleotide chain. Double helical means it contains two strands. Poly means more than one. It contains more than one number of nucleotide. In the year of 1953, two famous biologists, J.D. Watson and F.S.C. Crick, described the three-dimensional feature of DNA on the basis of X-ray crystallography, and that model is famous as Watson-Crick model or double helical structure of DNA model. In that model, they describe how this pentose sugar, nitrogenous base and phosphate group arrange to form this DNA molecule. What this Watson-Crick model describe? DNA molecule consists of two polynucleotide chain. They twisted around each other around a common axis in right-handed way to form right-handed double helical structure. Suppose here I am making one axis. This is a axis. Two polynucleotide chain. This one is a nucleotide chain. This one is another nucleotide chain. They twisted around each other. They turn around each other around a common axis. In right-handed way, in this way, they turn around axis. They turn in such a way that they both are anti-parallel with each other. Suppose one will run in this way, other will run in opposite direction. If one will run in this way, other will run in opposite direction. So what will happen? 3' end of one strand will face towards 5' end of other strand. In similar way, if we look the other end of both the strand, this one having 3' end because here is 5' end, and this one having 5' end. Both will face towards each other. And each having nucleotide, one will have, if purine base, other will have pyrimidine base. Suppose this strand having bases like adenine, guanine, cytosine, thymine. Like this, other will have thymine, cytosine, guanine, adenine, thymine, thymine. If one will have purine base, at the same place in opposite strand, you will find pyrimidine base. And adenine will bind with thymine by 2 hydrogen bond. These bonds are hydrogen bond. And similar way, guanine will bind with cytosine by 3 hydrogen bond. A will bind by 2 hydrogen bond with thymine, and cytosine will bind with guanine by 3 hydrogen bond. This is the base pairing rule of DNA. So C will bind by 3 hydrogen bond with G, A will bind by 2 hydrogen bond with T. So what will happen? Both the strands will remain stable by hydrogen bond. If you see its structure, you will find the nitrogenous bases will present at the core position of this DNA, and the sugar phosphate backbone will remain outside, or you can say both the side of this nitrogenous bases. Both the side you will find the sugar phosphate backbone. It will also follow another one rule, that one is Chargaff rule. What this Chargaff rule will say? The number of adenine you will find in DNA, it is equal to the number of thymine. It's right, because it's A will bind with this thymine, and the number of cytosine you will find in DNA, it is the similar number with the guanine present in DNA. And the summation of this purine base is adenine and guanine, its summation is equal to summation of pyrimidine base. Pyrimidine base is cytosine and thymine. These are the pyrimidine base. Both the summation are equal with each other. So adenine and cytosine, if we will add, it is equal to guanine and thymine. And the ratio between this adenine and thymine with cytosine and guanine is constant. It will vary from 0.4 to 1.9. If we see the diameter of DNA, this is the diameter, this part. This is the thickness of DNA. This diameter is nearly 20 angstrom. 20 angstrom. The distance between two bases, two consecutive bases, this is one base, this is one base. The distance between this part is nearly 3.4 angstrom. If we see the pitch, or you can say torn, this is 1 ton. 1 ton means it will rotate 360 degree. Will be the rotation. So how you will calculate? Suppose this one will start from here. These two up to this, it is a 1 ton. This is known as pitch. This to this. It's having distance 34 angstrom. So in one pitch, or you can say in 1 ton, you will find how many bases? The distance between two consecutive bases is 3.4 angstrom. The distance of one pitch or 1 ton is 34 angstrom. So there are 10 base pair present in one pitch, or in 1 ton you will find 10 base pair. In some place you will find somewhat little deepness, and some part you will find somewhat deeper portion. This is very small deep, this is somewhat larger deep, or you can say deeper portion. This deep group, or you can say major group, is nearly 2.2 nanometer, and this shallow group or minor group is nearly 1.2 nanometer.